Hello, so my name is Ken Yokoyama, and I'm from the University of Illinois in Champaign-Urbana. And today I'm going to be talking about constructing sequence similarity networks, what they are, how we construct them, and what they're good for. And at some point I'm going to also mention why it's necessary to use blue waters in order to do what we do, because it's very necessary in our work. But first, I'm going to try and answer three basic questions. So first, who are we? Second, what is it that we actually do? And third, what are our goals in terms of serving the scientific community? So first, who are we? So our lab is dedicated to producing and maintaining the Enzyme Function Initiative, so EFI for short. And this is designed to be an interactive web tool to help biologists study protein function. So what do we do? So the idea is this. So there are lots of biologists that study protein function, and there's a very good reason for this. So every chemical reaction that occurs in the body or any living cell is carried out through a protein or an enzyme. And since there are lots of different kinds of chemical reactions, of course there are a lot of different kinds of proteins, and so a lot of them have different functions. Here, if you look at a protein, here we have a transcription factor from E. coli, so bacteria, and here is a protein in humans called hemoglobin. And as you can see, these two proteins look very different. And as a result, they do extremely different things in the cell, right? So this regulates how much protein is made of certain proteins within bacteria. This simply carries oxygen throughout our bodies as humans. Now, since they look so different, of course, they have very different functions. However, if you look at, say, this protein, and you'll have to kind of excuse the illustration. So these two proteins are about the same size, but if you were to look at these two proteins closely, they look very much the same. And so this is because this is hemoglobin in mouse. And so it essentially does the exact same thing as hemoglobin in humans. It just carries oxygen throughout the body. Now, what if we were to have some kind of protein where we don't know what it does? And so there's a vast number of proteins out there that are known proteins, but we just don't know what they do. So our goal is basically to differentiate pairs of proteins. So for instance, we can tell that these proteins are similar, whereas these two proteins are not. The idea then being, what are our goals? Our eventual output looks something like this. So this is a sequence similarity network. So what you're looking at is each of these nodes is a protein, and each of these edges between the two nodes essentially says that these two proteins are similar to one another. And so we can tell that this protein is, for instance, similar to this protein, but it's actually not similar to this protein, and so forth. So this is the eventual goal. And so the idea then being is that if, say, this protein, we don't know what the function is, then we can make some kind of an educated guess as to what the function might be based off of, say, this protein might have a known function, and so forth. So the background is, how do we do this? Then protein function, this is what we're interested in, is protein function. Now, protein function is determined upon protein structure and the chemical characteristics of proteins, of the protein, and this is in turn dependent upon the amino acid sequence. This is in turn generated by the genomic DNA. Now, we don't look at the genomic DNA, but of course we look at the amino acid sequence. So how do we get from the amino acid sequence to the protein function? This is actually not a very easy question to ask because, or to answer rather, because if you look at the amino acid sequence, it looks basically like this. It's just a long string of letters, right? And so how do we get from here to protein function? This is very difficult, especially because for most proteins, we don't have the structure, but we do have the sequence. And so it turns out that the only way that we can do this is essentially to compare it to other known proteins. So if you were to compare it, it would look something like this. So this is the output from BLAST. And what you're looking at here is these top rows are the amino acid sequence of the ones that I just showed you. This is a comparison to a different protein. And so in the middle, you can see each time that you see a letter here, there's that means that the amino acids are the same between the two sequences. Now you can see that in a lot of cases there are similarities, so these two proteins are not very dissimilar, but they're also not identical. So we're looking for a way to quantify how similar these two sequences are. Of course, BLAST has a way to do that. So this is the bit score, and I'm not going to go into this too much, except for, so each of these terms represents the bit score for a single column in the alignment, and this is essentially just a likelihood ratio, log likelihood ratio. So comparing the idea that these two are similar versus these two are just made of random amino acids. And so most people are, if you run BLAST, most people are more familiar with the e-value, but the e-value eventually is derived from this score. So we take this score and we make a second score, which is an alignment score. This bit score here was the score that I was just showing you. And the idea here is that the higher the alignment score, the more similar the sequences are. So you can set some kind of threshold, and every pair of proteins that has an alignment score over this threshold, then we draw an edge here. And so you can see the clusters, so all of these proteins are similar to each other, and all of these two are similar to each other, but they're not similar to these, and so on. It's fairly simple, except that you can, of course, this is supposed to be interactive, so you can change the score. And so the more lenient the score, the more edges you get. The more stringent the score, the more edges you lose, and so on. Now, what's the problem with this? Well, the number of known proteins is exploding, okay? And so, I mean, basically all of this is not difficult to do, except that the number of proteins is so unbelievably large. So just, this was last month, there were over 62 million known proteins. About half of these have unknown function. Just last year, you're looking at just under 48 million. So this number is increasing by a whole lot every year. Now, the difficulty is, now, how do you deal with all of this data? Well, there's an interesting database called Pfam, and this database has protein families which essentially group together proteins that have similar biological functions. The protein families are based off of sequence similarities and hidden Markov models. The only thing about Pfam is that, of course, you get these huge families, but you don't necessarily know which sequences are similar to which other sequences within the family. Now, 50% of these proteins have unknown function, again, so there's a bunch of them that we just don't know what they do. The family sizes, so this is what makes it difficult. So the largest Pfam family contains over 800,000 total sequences. Okay, so 63 of them contain over 100,000 sequences, and 186 families contain over 50,000 sequences. In total, there are 16,000 families, so you're talking about a huge number of sequences. Now, what we're doing in general is pretty simple in theory, except that once you start to realize how large this is, so you're having to do every single pairwise comparison, right? So it's going to be n choose 2 comparisons. So that's on the order of n squared comparisons, right? So just in order to do this one single family, you still have to break it up into smaller bits and then do every pairwise comparison and then concatenate them all together. But even if you do that, then even if you break it up into, say, we break it up into 64 different pieces, to run BLAST on each of these pieces takes over a day. And so just doing this one family would take a really long time, especially because Pfam gets updated every 2 months. So if we're to try and actually do this in a timely fashion so that we can get the user the most timely data, then we have to basically run all jobs simultaneously. So how do we deal with all this data? Essentially use Blue Waters to run jobs simultaneously. The idea is to construct sequence similarity networks for each Pfam family. And in the end, again, this is what we get. And this is a smaller family, but of course for the larger families, we get, this can look extremely complex. And the idea, again, is this is mostly just, it's a simple idea, but it's very helpful for biologists who might not be, computationally not be interested in the computation, but it is helpful in terms of, so if you want to know what this protein does, then how do you determine that? Well, one way is just to see what its neighbors are and so forth. And this is very important because, like I said before, about half of all known proteins have unknown function. So there's just there's a wide range of proteins that we just don't know what the function is. And so this helps a lot of biologists try and at least make an educated guess as to what their favorite protein does. And I'll take any questions or comments.
So it looks to me as though you might be overlooking something that could be very helpful to you. Mm-hmm. And this is to break your proteins down into their constituent domains. Mm-hmm. And then do a characterization of families based on domain composition. And then you only have to do your full protein-protein comparison in proteins that have the same domains. And especially this is because your BLAST may be very unreliable because it depends on an alignment mm-hmm. that may be wrong if you have strayed too far away from the same domain composition. So,
can I make a?
Sure.
So Pfam actually has domains itself.
Yes, Pfam does have domains, right.
So this is the data that we use. So it's not necessarily the entire protein. In fact, Pfam itself can have, in the same family, multiple segments from the same protein as different sequences. Does that make sense?
Yes, but in the very large Pfam families that you've mentioned, mm-hmm. There's a great variety of domain compositions within the family. Mm-hmm. You could characterize those domain compositions with the program MEME. Are you familiar with MEME?
Yeah, yeah.
Yeah, okay. So there's a way of going back and forth with MEME and MAST that I think may give you both more detailed and perhaps more reliable information than you're getting now. I'd be glad to, since we're at the same institution, I'd be glad to talk to you about it
Right.
at more length, but I think that this might be useful to you. And yes, of course, I agree. Pfam, the domains are implicit in your use of Pfam, but I think that you can get more detailed information by actually independently looking at domains.
Right. So one thing that we had thought about, or at least that I had thought about, is to use hidden Markov models to more differentiate between. So for the larger families, how can we separate these into smaller subfamilies?
Right.
So I think MEME would be a something that's out there that's already. My understanding is that that's mostly for transcription factor binding sites. Is that correct, or?
We have used it extensively for coding regions.
Oh,
so
really?
You can do that.
Oh, okay.
Yeah, I mean, it's great for transcription factor binding sites because it's the only thing, because you can't align them at all.
Right.
But you gain something by using it for coding regions, and the reason you gain something is because even though you can align them, sometimes the alignments are kind of flaky.
Oh, right. I see. Okay.
Okay, good. Yeah, let's talk more.
Yeah, definitely.
Wow, it's time to great initiation.
So in the same vein, if the problem is the growth in the number of proteins, presumably you don't have to recompute the previous interactions. So if you add one new protein to you've got 100,000 already done, then you've just got to do an n-way comparison. Or am I wrong?
In theory, we could do it that way. Sometimes they, so we would have to check which ones are new.
Sure.
I mean, so that's actually a good point. That the only thing is that for the largest ones, so it's also sort of true that sometimes the families themselves break apart, and so doing this on a large scale, they might add families, they might make one family into two families, they might make two families into one family, and so on. Now that being said, so part of the problem would be is that it's not, like I was saying a little bit before, so these are not necessarily full proteins, but they're also modules. So the sequences themselves can differ by a little bit, and so it makes it a little bit more tricky to do what you're suggesting, but that actually might be one thing to look into. So yeah, thank you for that's something that we hadn't really thought of. We're just doing it mostly brute force, but yeah, so.
Any other question? So if you had even more compute power available, what sort of things would you be able to do anything more with it? What's your next sort of step?
What I would actually like to do is build phylogenies, rather than just doing the pairwise comparisons, to build phylogenies based upon, sequence similarity, to build trees. That's one possible thing that we could do, but that would take a lot of compute power. So.
Okay, so thank you for sharing your research progress with us. Can you briefly introduce what is your most important progress this year done by your research team?
So the most important one is that we actually got that large family done, which has never been done before. So that was our big thing, so that we did actually get the all-by-all blast accomplished using blue waters. So that was, for us, pretty big.
Okay, sounds great. Any other question?
Mm-hmm.
How do you connect blast to blue waters? I mean, I guess this is an interactive thing, or you run it on the database? I mean, so
Say
you,
it again?
So you use the API with the website of Blast, or you use the Blast program?
No,
and
we
you
use the
run it
Blast executable.
executable.
Yeah,
program?
yeah, on
And
blue waters.
How does it scale on blue waters? I mean, this
I mean, it works well. The only thing is that, you know, I mean, it's just a command line thing, except that you're running a whole bunch of jobs simultaneously, right?
Okay.
So there's shell script after shell script of what exactly you're running. So you want to run this Pfam family, and then within this Pfam family, you want to run 32 per, you know, however many. So
to appendix question, you run it on CPU, right? I mean, it's not a GPU-enabled code.
right.
And you take only one node for each.
It depends on how big they are. So some of them only take one node. Some of them take, for the largest one, we take 70.
70, okay.
Yeah.
Great, thanks.